how are you seeing, right, as a consultant working with clients, how have you seen some of these guidelines, some of the work being done analytically going on to inform or the industry's process development work? Is it, right? How are you seeing it
It,
kind of being used practically?
it is starting to evolve a lot. So, and part of it is the process development now asking the proper questions, like other
Mm,
modalities. Is the method appropriate to tell me my process? Let's say it's getting rid of all double-stranded DNA.
mm,
And is the method that we have on hand capable of detecting nothing, detecting something? How does that relate to my process? The other thing is everybody, was doing, label it as a whole cell protein, but we claim that mRNA is synthetic. So you're really not using cells. What you're using is enzymes. So now the question is, are these methods that being used appropriate to test for the clearance of any enzymes, whether it's polymerase, RNA polymerase T7, or other enzyme that you're using for the polymerase, or if you're doing enzymatic capping, is that enzyme cleared? Does your method detect that enzyme at a level that's needed? So now you start peeling the onion and start getting more into the little details, and
mm-hmm.
this is part of evolving drug development, and the process development folks gonna need that, and they're gonna need a reliable method to tell them that my process is capable of this, and the proof of it is that the methods that we're using can tell us that.
That's beautiful. Yeah. We're really, as we continue to learn more, just there's so many more nuances that we have to wrap our heads around, right? So
Absolutely.
that's the fun part. That's the fun part. So I want to turn to Jeff and Jim, right, to kind of build off of what Khaled was saying here. You know, first of all, starting off with, the knowledge that we have right now in terms of linear mRNA, right? So Jeff, I know you guys are working with linear mRNA. How have you seen sort of the analytical developments in that space, right? And for the vaccines as well, right? How have those been influencing the work that you're doing with your process development teams today? Are you asking the right questions, as Khaled would say? Dangerous
I
question.
hope. First of all, thanks for having me on, and yeah, really looking forward to the conversation. And so the answer, yeah, I hope we're looking at the right things, right? That is part of an evolving field, is to try to figure out what the right things are to look at. I think we have a great advantage in the therapeutic area in that the vaccines really cleared the way, and a lot of the standards were put out there. So we have the opportunity to build on top of that. What's different is, the therapeutics, it is different. Like the doses are like 1,000 times greater, right? Like there's a whole lot of stuff that now becomes maybe more important than it was in the CQAs for the vaccines. A different focus on some of those analytical methodologies. But in the end, those analytical methods, like we need to continue to improve them and come up with new methods. That's probably, I'd say, the most difficult, the biggest challenge in the field right now is we're still, it's a little bit like looking through a foggy window. We don't know exactly what we're looking at yet. There's still a lot of questions on, what is it that I'm seeing? How important is that? When it comes to like fragments, right? Like how, from an integrity standpoint, what and is not going to cause harm, but will be efficacious as well. So I think, if I were to put one method out there, that's, kind of the integrity and understanding the integrity of the RNA is really high up there. Probably the other one that I think is a continual challenge is the potency, right? Understanding potency. Those are the two biggest challenges right now, and we're seeing a lot of improvements in methodologies and instruments and crossover from previous things. But those are the areas that still need a lot of work has come out. You know, there's been a lot of great work
Yeah.
in those areas, but I think we're still lacking that understanding.